This video tutorial presents GenoMiner, a tertiary analysis solution for next-generation sequencing data. Current applications can be used for reference and de novo assembly as well as ChIP-seq data analysis. In the video, we are going to demonstrate how easy it is to work with GenoMiner. GenoMiner can work with genome reference files in FASTA format and NGS result files in a format relating to the sequencing technology. The source of the data can be any Illumina, ABI Solid, or Roche NGS machine. This video presents how GenoMiner manages the analysis of ABI Solid reference assembly dataset. The first step is to create an experiment and import the files you want to analyze. If you need to run an application on a previously used file, you have the possibility of choosing one of the former generated experiments. Let's create a new experiment titled Demo Experiment. At first, the files should be imported. We need a reference genome in FASTA format, human FASTA. We have found it essential to give the possibility to rename the input and reference files on demand. The upper window on the left lists the successfully imported files. As we can see, the file is designated with the name given by us and the type of the file. Then files containing the reads CS/FASTA and quality values qual are needed. We follow the same protocol as before. Now our file imports are done, the files for analysis are listed here. After importing the files, it's time to choose the method of analysis. This is what we call a workflow. Name your workflow carefully, so it will be easy to recognize later. Our workflow had the name ABI Reference Assembly Test. The next step is to define the modules of the workflow you have chosen and set their parameters. Modules are the tools of workflows that are used for error correction, reference and de novo assembly, or ChIP-seq data analysis. The next step is called error correction. If you want to run GenoMiner with Illumina or Roche sequencing data, you need to skip this module step. As we work with ABI solid files in this example, we click on SAET to perform the error correction. Before setting the parameters of the error correction tool, the system displays the files we intend to analyze further. The aim of this simple step is to make sure the user has chosen the correct files. Furthermore, it provides an opportunity to change files as needed. The input of an SAET error correction tool consists of solid reads in CS/FASTA format, a file with quality values in qual format, and an expected length of the sequence, from which reads were sampled. As a first step, a spectrum of all k-mers is constructed from the set of all reads, and then each read is corrected one by one if necessary. SAET transforms corrected reads and quality values into a new CS/FASTA and qual files respectively. Behind the question marks, you will find some help about setting parameters of modules. The information provided here can navigate you through the parameters given the right setting for your particular analysis. All parameters have a default value, but they can be adjusted according to the aim of the experiment. The next step is to find the right reference assembly tool for your dataset. Both BWA and shrimp support letter space as well as color space reads. Let's continue with BWA. The pop-up window shows the genome we have uploaded as reference genome. Here, the user had the possibility to change it as well. For indexing reference genome, we choose the option BWTSW, which is for large genomes. All other parameters stay as default. Click Save Parameters button to promulgate changed options. After compiling the workflow, we can start it immediately by clicking the Save and Run button, or escape the whole workflow by clicking Cancel. Alternatively, we can save our workflow without running it straight away. A smart option enables you to set the system to automatically send an email notification once the workflow has been finished. To take advantage of this option, give a valid email address. We can trace the process of analysis on the user interface. Click the button on bottom right-hand corner to hide this window. The left side box shows the workflows being underway. This reference assembly workflow takes about 2.5 hours to run. After receiving email notification that workflow has been finished, we can open the experiment folder and take a look at the result files. The BAM file can be opened with GenoViewer. It is a powerful and comprehensive browser for the SAM-BAM format and GFF annotation files commonly used in the assembly tasks of next-generation sequencing data. GenoViewer is a freely available product also developed by Astrid Research Incorporation. After opening GenoViewer, import the reference genome in a FASTA format. Throw a glance at the chromosomes listed on the left-hand side. You can scroll each chromosome with the scroll bar. Then open the BAM file. The alignment file also contains more contigs. Always remember to change the appropriate chromosome in both the reference genome and the alignment file. Scroll through the chromosome to click the aligning reads, mismatches, SNPs, and indels. After clicking on a read, a pop-up window displays the alignment of the selected read to the reference sequence. Options displayed in the bottom left corner of the window can help you find the SNPs, insertions, and places of deletions. GenoViewer allows you to generate the consensus sequence and mutation tables with all the deleted polymorphisms and indels and then export them into a new folder.